चैप्टर 4 में एक टॉपिक तो हो गया है. I am sure biosynthesis हो गया है, but I am not sure whether molecular machines part is done or not. Please let me know if it is done or not. Molecular machines. I'll just share the presentation. You just look at it and tell me whether it is done or not. Yeah, see, I'm presenting. Did we complete this part?
Okay.
It's been 1 week since I have taken a class. So I just want to know what was the last class I have taken. Is this topic done or not? Somebody please tell me if it is done or not.
As far as I remember, we haven't done this part.
No, sir.
We haven't done this part. Okay, so we will do this part today. And tomorrow we will be completing the course. Tomorrow we will complete chapter number 5. Okay. So if you're going to look at a cell, the cell itself is kind of a miniature manufacturing industry where you're going to have a lot of units. Like you have a manufacturing unit, you have a waste management unit, you have janitors, and then you have so-called retrieval units, etc., etc. Right? So if you're going to look at the miniature level of the cell, you are going to have a lot of molecular machines that are actually running across the cell, doing a lot of mechanical activity. All right? And this way, the creation nature itself is a wonderful creature. So if you're going to look at the molecular motors that are available in our cell, we have different types of molecular motors. Some of the motors are protein based, some of them are DNA based, some of them are RNA based, some of them are pure chemical based. Example, look at the motors that are available in your RNA assemblies, like your so-called spliceosome complex, the mRNA editing. mRNA editing is done by self-assembling proteins, which assembles and then which makes the exons paste and which makes the introns to remove. Okay, so they identify a particular region, they cut the RNA, and they patch up the RNA which is required. The same way, you look at the translationary machinery. Translation machinery, maybe you have 3 different types of RNAs acting for a synthesis of a protein. Right? All of these are self-assembling; they are automated. Same way, we have different protein-based molecular motors available. Some of them take part in rotary activity, some of them take part in linear activity. So we are going to study about only these 2 types of motors. One are rotary motors and the other one are linear motors. Coming to the rotary motors, these are the motors that basically are dependent upon the chemical gradient. So if you're going to come across the chemical gradients, we are dependent upon 2 major ions. One are hydrogen ions and the other one are sodium ions. Okay, so hydrogen ion concentration is usually coming out through the metabolism. So we have a series of reactions called as electron transport chain in the metabolism, through which you tend to have a higher concentration of hydrogen ion. And this higher concentration of hydrogen ion will be generating a force called as proton motive force, PMF. The PMF will be driving a rotor located in your membrane of the mitochondria called as C12. So this rotor is the one which gets moved. Okay, so it undergoes an angular momentum, and the angular momentum will be carried downward through a stalk, creating a torque. And the same torque is utilized by the inorganic phosphate to couple to a adenosine diphosphate, leading to okay. So if you're going to look at this molecular motor, these are the fragments. You have 2 fragments. One is F0 and the other one is F1. F1 is located inside the mitochondria. F0 is located on the mitochondrial membrane. This is the inner membrane. Okay. So this is the one which is rotary in nature, and this is the one which is static. Basically, this also exhibits a rotational force, but that is very rare. This is the one which is most commonly exhibiting your rotational moment. Okay. So if you're going to look at the combination, what are the factors available? The lower rotor F1 basically is made up of alpha. We have 3 units of alpha, 3 units of beta, 1 gamma, 1 epsilon, 1 delta. So these are the components of your so-called lowermost rotor F1. And coming to your upper rotor, this is made up of three different components. B component, which is usually required for adhering onto the peripheral supports. Then comes A component. A component provides inlet and outlet of the hydrogen ions. And these is very crucial for rotation of the C complex. The C complex is made up of proteins which range from 12 to 14. Okay, in case of mitochondria, we have 12. In case of chloroplast, we have 14. In case of bacteria, also you find a similar motor available, and that will be very crucial in generating something called as ATP. Okay, so how does the mechanism happen? How does this motor basically function? Let me share a small video clipping so that you'll understand. Yeah, so look at this clipping. So this is your molecular motor, this is your mitochondria. So if you're going to look at the mitochondria, we have a series of complexes at the inner membrane of the mitochondria. These are called as, these usually takes parts in electron transport chain. So as you can see here, you have these many number of complexes that takes parts in electron transport chain after the Krebs cycle. So once we get the products, these products are undergo oxidation, through which most of the electrons and most of the protons gets carried forward into the space available between the membranes. And this creates a higher hydrogen ion concentration in between the membrane space, creating something called as proton motive force. Now, at the end of each complex, you have this motor called as F0, F1 rotor ATP synthase. So what is the importance of this ATP synthase? You can see here we have freely available adenosine diphosphates and free phosphates. These usually will be associated to one of the lowermost compartments where they undergo coupling process. So to undergo coupling process, you require a flow of hydrogen ions. See, all of these basically exhibit reduction and oxidation. So first they get reduced, followed by they get oxidized. So you have an inlet for entry of hydrogen ion, and you have an outlet for exit of hydrogen ion. So entry of hydrogen ion will be from the membrane space, and exit will be into the matrix of the mitochondria. So just look at the way एक बार reduction हो गया, फिर oxidation हो गया है. So the oxidized part should be now moving into the inlet. So you undergo an angular momentum of 30 degrees. And this way, approximately 120 degree of angular momentum will lead to a generation of enough energy that makes this inorganic phosphate to bind to the double phosphate available here, making it a triple phosphate structure. And that's how we are going to get ATP in the mitochondria. ओके सो दिस गोज ऑन मतलब यहाँ पे एक बार हो गया है अब यहाँ पे देख लीजिए यहाँ पे बाइंडिंग हो रहा है फिर यहाँ पे बाइंडिंग होने के बाद पीछे वाला ब्लॉक है वहां पे बाइंडिंग होता है सो दिस वे फॉर एवरी फोर simultaneous oxidations and reductions you're going to have a generation of ATP. This is in case of mitochondria. So this is how your ATP generation happens through molecular machine, which is rotary in nature. This is the smallest rotary machine available in the nature. Coming to the larger category of rotary machines, so the larger category of rotary machines is exhibited by your so-called structure called as bacterial flagellum. So if you're going to look at the bacterial flagellum, unlike this basically have only two rotors, and out of this one rotor is only rotating, other one is static. Here is not the case. All the rotors that are available here will be rotating. And if you are going to look at the bacteria has multiple layers, right? They have a membrane followed by cell wall, followed by capsule, etc., etc. So they have many layers on the top of the surface. So each and every layer has its own rotor, and surprisingly, all of these rotors are interconnected by a central rod, right? So all of them tend to rotate in a synchrony. मतलब ये अगर clockwise direction में rotate हो रहा है तो automatically ये भी rotate होता है और ये भी rotate होता है. And same way, this entire complex of rotors with the rod is called as a basal body. Followed by that, you have a hook, From the hook, you have auto assembling complexes coming out to make a long wig-like structure called as flagellum. So this is how a bacterial flagellum look like. See this component. This component is the basal body, which is made up of all the rotors and the central connecting rod. Followed by we have a connecting hook, which is usually bent. So this is made up of structural proteins. It also undergoes the same rotation for which is exhibited by this. And following that, you have self-aggregating proteins called as flagellins. The flagellins will be making this entire whip-like structure called as flagellum. Okay, so if you're going to look at the regulators here, the regulation basically happens through this charge, positive charge. Okay, so the positive charge generation happens either through hydrogen ion concentration or through sodium ion concentration. And you have two proteins which takes part in making this rotors to rotate. One are mode proteins. You have two different types of mode proteins, mode A and mode B. So usually you find them in the lowermost rotors. The green ones that you're seeing here are the mode proteins. And followed by that, you have fluid proteins. Fluid proteins are basically required for switching of your so-called rotation. मतलब समझ लीजिए आपका rotation clockwise rotation में जा रहा है. Now we have to stop clockwise and we have to go into anti-clockwise direction. So the switching happens basically by these fluid proteins. Okay, so what are the what are the numbers? Let's look at the numbers. If you're going to look at a rotation, the speed of the rotation, it usually will be associated with approximately 150 millivolt of electrical potential that is generated usually because of hydrogen ion or sodium ion. And on top of that, when it is undergoing a clockwise rotation, the rotation is approximately like 1000 hertz. Okay, and a torque of approximately 8 to 16 is generated. Okay, and these mode proteins will be utilizing the torque, and they make this entire rotors to undergo either a clockwise rotation or a counterclockwise rotation. So whenever I'm talking, I was talking about two rotations. One is clockwise and the other one is counterclockwise. So when does the bacteria exhibit clockwise and when does the bacteria exhibit counterclockwise? Uh, whenever the bacteria is swimming, the bacteria will be exhibiting a clockwise rotation. Okay, and when the bacteria is static, matlab when the bacteria is sitting in a specific area, in that case, the rotation is counterclockwise. Okay, so let me just share a small video clipping from the YouTube. Yeah, look at this video clipping. So this is your anti-clockwise rotation. Okay, anti-clockwise rotation में speed काफी कम होता है because when the bacteria is idle, when it is sitting and when it is eating food, excuse me, in the vicinity, then only this anti-clockwise direction happens. And when the bacteria has to stop and when the bacteria has to run, the fluid proteins will stop its anti-clockwise direction and will activate the clockwise. So now in the clockwise direction, the speed is a bit higher. And so the bacteria swims across the media and will be moving. So the speed with which the bacteria travels is approximately more than like 50 times of its body length. And so like they are one of the fastest moving creatures on the earth. Okay, so you can see here rotors, जिस तरफ rotors rotate हो रहा है, same way the entire rod is connecting all the rotors and everything is moving in synchrony, including the hook and including the flagellum also. So look at the flagellum. The flagellum is exhibiting a spiral rotational motion. Once again, have a look at it. Yeah, as you can see here, see this is not a wave format. This is a spiral rotational format. All right. So that's how the largest of the rotary motors do work in the nature. Now coming to the linear motors. In linear motors, we have three different linear motors. One is myosin, the other one is dynein, and the other one is kinesin. So all of these three usually will be पहले के केस में क्या हुआ था? We have seen that these molecular motors are generating the energy. मतलब they are rotating, and when they are rotating, they are generating the energy. This generated energy is being showing some sort of a mechanical activity. But in case of linear motors, it's not the scenario. Okay, in case of linear motors, the scenario is different. So what they do is that they cannot generate the power on their own. Rather, what they do is that whatever the power is available in the vicinity is being utilized, and they will be exhibiting some sort of a mechanical activity. Okay. So before going to that, let's study about the types of bacteria depending upon the availability of flagellum. If your bacteria do not have a flagellum on its body, then it's atrichous. If the bacteria has single flagellum, it's monotrichous. If the bacteria has so many flagellum just on one side, then it's lophotrichous. If the bacteria has two flagellum only, but both of them are located on opposite sides, then we call it as amphitrichous. If the bacteria has many flagellum, but many flagellum are located at only 2 points of origin, then we call it as amphilopho. Lopho matlab tuft of flagella. Okay, so if you have two tufts of flagella, it's amphilopho. If you have single tuft, it's lopho. If you have many flagella all over the body of the bacteria, then it's peritrichous. So these are the different types of bacteria depending upon the availability and locations of the flagellum. Now coming to the superfamilies of cytoskeletal motors. So these are the linear motors that completely are dependent upon the cytoskeleton. So in the cytoskeleton, we have two elements, microtubules, microfilaments. Okay, cytoskeleton हम cell structure पढ़ते समय हम देख चुके थे cytoskeleton का role क्या होता है. They provide a rigid support to the cell, and on top of that they provide the framework. So utilizing this framework, there are some mechanical activities happening inside the cell. Okay, so we have three different types of motors, linear motors that depend upon them. One are myosins, the other one are kinesins, and the other one are dyneins. So these are the smallest proteins that are usually linear proteins that are available. So they cannot work independently, rather they work in the groups. Okay, whereas these two, kinin and kinesin and myosins, they work independently. Okay, so what does the myosin do? All of these three have a common structure. You can see these yellow color globular heads. These are the motor domains, which generate the so-called mechanical movement. Okay, the mechanical movement generation happens because of breakdown of energy molecules, ATP. And followed by that, you have a stalk. As you can see here, it's written stalk. This is the stalk. The stalk basically at the end is flexible. Okay, so these are made up of a few chains of amino acids. And towards the lower end, you may or may not have a light chain. The light chain is basically for connecting onto the so-called cargo or to any other structural element. Okay, so first we'll begin with the myosin motors. So myosin motors will be working on these filaments called as microfilaments, which are made up of actins. So actin, you see, this is how the actin dimers will be formed in the cell. So these myosins usually will be making the movement towards the positive end. So what do you mean by positive end and negative end? Whenever a cytoskeletal element is established and whenever it is constructed, you see there is a point of origin and there is a point of growth. The point of origin is always negative and the point of growth is always positive. Okay, let's say this is your point of origin of this microtubule or microfilament. So this is your negative end. And let's say this is the growing end. So this is positive end. So the movement of actin filaments always myosin to actin will be always from negative to positive end. Okay, so how does the movement happen? You have two different kinds of strokes. One is a power stroke and the other one is relaxation. Okay, हमारे muscles में we undergo a sort of movement called as contraction and relaxation, right? So if you're going looking at contraction and relaxation, as you can see from this is okay. So look at this. Your muscles can actually undergo contraction and relaxation. When they bind, they will drag these actin filaments towards the positive end. So you can see now the adenosine diphosphate molecule will be released, and that provides the power for the mechanical activity. And this is the mechanical activity that is exhibited. So this state is contraction. This is your contraction state. So all of your muscles here now undergoes contraction. The volume of the cell is shrunken. Once this is done, then another ATP molecule will come in, and the binding of ATP molecule will make the myosin to release. And here we are undergoing a relaxation. So the muscle now undergoes relaxation. So this way we have a simultaneous contraction and relaxation happening, and they are completely dependent upon something called as your microfilaments, which are made up of actins. Okay. Coming to the second category of linear motors. As you can see here, these are the kinesin linear motors. So what they are doing is that they are basically acting as coolies of the cell. Coolie matlab porter. Okay, the one that you find in bus trains or railway stations or anywhere else. So what they do, they carry the large amount of cargo on their head, and then they will be traveling from one location to other location. Okay, they deliver the cargo basically. So in the cell also we have these vesicles, packed contents to be delivered from one area to other area. So we have different modes. One of the mode is through kinesin transportation. So what does this kinesin do? The motor domain will be completely dependent on this is your microtubule. Okay, and if you're going to look at this fellow is your motor acting fellow called kinesin. So see, you can have a kinesin working here, you can have a kinesin working here, here, here. So at multiple spots, you have the cargoes being delivered. So see, here we have one of the fellow doing the job, and here this fellow is doing the job. So if you're going to look at the structure, this is your stock, this is your light chain. The light chain is bound to this cargo now, which is very, very big. And this is your motor domain. The motor domain is dependent upon the microtubule. So it moves from a negative end to a positive end. So you can see these are actually walking. So how do they walk? The motor domain will be spending 1 ATP, and for every ATP, the stalk makes a movement of approximately 80 angstroms. And this way, because of the flexible stalk, the motor activity is done, and that makes the movement to happen on the microtubule from one location to other location. So this is about your kinesin linear motors. Coming to the third category of linear motors called as dyneins. So dyneins, as I said earlier, they cannot work independently. Rather, they have to work in the group. So if you're going to look at this is your eukaryotic flagellum. Prokaryotic flagellum or eukaryotic flagellum का movement देख लीजिए काफी अलग है. This is exhibiting a wave motion. This is not spiral. Prokaryotic में spiral था. ये यहाँ पे eukaryotic में wave motion है. So how is this wave motion being generated in the flagellum in the eukaryotes? So here we have this interconnecting microtubular. You see, these are the microtubules, which are double structured microtubules. All of these are connected by these motor proteins. These are the motor proteins. As I said earlier, they cannot work independently, rather they work in the group. So all of these motor proteins act at a time on one side. Okay, so they make the microtubules move like this, and this generates something called as a power stroke, making it to take a forward bend. Okay, forward bend matlab ek trough. In a wave, you have a crest and a trough, right? Or you have an amplitude and you have a depression. So when a power stroke is happening, we usually get a amplitude or a crest. Okay, so these things when they are working, when as soon as a crest is being created or as soon as an amplitude is created, you see these particular segments will show inhibitory signal, because of which they don't function for some time. Okay, and in this way, if you're going to look at the cross section, see, we are having different color codings. These are in blue and these are in somewhere in orange. Okay, crimson orange color. So the blue ones are the active ones and the orange ones are the ones which are undergoing inhibition. So on the entire structure of a flagellum, at one side you have a generation of forward bend, and at the other side you have a generation of reverse bend. So wherever reverse bend is generated, these dynein motors are undergoing inhibition, and wherever a forward bend is happening, there the dyneins are basically actively functioning. So look at the functioning. See, these are working and these are idle. Same way, now these are working and these are idle. Now these are working. Now these are working. So when they are working, you can see there is a generation of wave. See, this is the forward bend. Once the forward bend is done, they get inhibited. So this way, alternatively, on both the sides, you have a forward bend happening and you have a reverse bend happening, and that makes the generation of wave. So you have a crest and a trough. So this way, the cilia or the flagella of eukaryotes starts beat. Okay, so they beat, they show this vibrational activity. This animation is showing in a slower motion, but usually when you actually see it in a real time movement of a sperm or any other thing, they move at a very fast rate. Like take our intestine. Intestine has cilia and all. So the cilia basically beats very fast. Okay, so these are the linear motors. These are some of the linear motors that perform the activity in our body. Okay, so that's all about molecular motors and machines. I'll share the slides with you all. And moving on to the next topic. The next topic is about nervous system. So what we can do is that we can start with some introduction because the topic is a bit bigger. So we can start with that introduction part. And where is this? Yeah. And the main functional part, we can do it in the next class, maybe tomorrow. Okay. Just let me know if my slides are visible to you all.
Yes, sir, visible.
Okay. So we are going to study about one of the most complex systems of our nature, that's nervous system. So why we are reading this nervous system? Because this is like one of the most important systems that actually led to the advancement of the technology. Right? We are dependent upon something called as networks, neural networks, and the neural networks are nothing but they are mimicking of our natural neural networks. Right? So a brain, it's very complicated. The kind of the neural establishment that happens in our body is very complex. It is very sophisticated. Right? People still did not understand how this networking actually, etc., has happened. But if you're going to look at the ability or the way that they perform the task, though we have millions and millions of neurons and though we have some trillions of connections available in our entire body, these all connections make the work happen in a very fast and a precise manner. Right? So it is important to study about how this organization has happened, and we are going to study about the preliminary cells that are available in the nervous system. As soon as people think about nervous system, the first thing that comes into their mind is about brain, and the second thing that comes to the mind is about neurons or the nerve cells. Right? Apart from the neurons, you have different other cells in the body which exist in the nervous system, and we call them as glia, GLIA, glia. So what are glia required for? They are basically the supporting cells of the nervous system. When the neuron nervous system is developing, all the growth, all the defense-based activities, and all the support, mechanical support, and everything is provided by these type of cells called as glial cells. So let's look at each and every one of them. So we have 6 different types of glia in the entire nervous system. The first one are oligodendrocytes. So they, what do they do? As you can see here, oligodendrocytes are projecting themselves, and the projections are ending up with a lipid-rich sheet called as myelin sheet, which provides insulation on the conducting area of a neuron. Okay, usually our neurons exhibit electrical impulses, right? They show you electrical firing. So when an electrical firing happens, it should not be leaky. At the same time, it should not affect the other structures. So you require an insulation. The insulation is provided in the central nervous system by these structures called as oligodendrocytes. Okay, and in the peripheral nervous system, peripheral nervous central और peripheral क्या होता है? Central मतलब brain और spinal cord. ये दो structures को मिला के हम central nervous system कहते हैं. इसके अलावा जितना भी nervous system है, whatever the nervous structures are there, they are peripheral in nature. Okay, so at the peripheral nervous system, the same insulation activity is done by another category of cells called as Schwann cells. So what are these two cells doing? Basically, they are providing insulation by forming a lipid-rich sheet called as myelin sheet. Okay, oligodendrocytes can make as many as like 14 to 15 projections. Usually, they will be like the range will be from 4 to 15 projections, and that's how they can cover the projections on more than two number of cells. So as you can see here, see this is covering up this cell, this is covering up this cell. So 1, 2, 3, 4, 5, 6, approximately 6 to 7 projections have been there. Okay, that's about oligodendrocytes. But Schwann cells, on the other hand, they are not like that. They will be able to make only one sheet, and they themselves will be sacrificing their entire body for making up this sheet. Okay, so this way, uh, in both peripheral and central nervous system, you have the insulation coming up from oligodendrocytes and Schwann cells. Coming to the astrocytes, why are we calling them as astro? Because they are star-shaped cells. They are the most important cells of the glial cells because whatever the movement is coming up from your circulatory system, especially the blood, out of the blood, it will be giving only few selective compounds to the brain. Other compounds are retrieved back. मतलब ये blood और brain के बीच में एक bridge establish करते हैं. Why do we require a bridge? Because uh, in our blood cells, in our blood, usually we have so many number of cells giving so many number of factors. Each and every factor has a chance to give a super big noise to brain signaling. मतलब brain का signaling जो है वो minute factors also can disturb the brain signaling. So to avoid these many number of factors that are available in the blood, you require a bridge or a barrier to get established. And the establishment of the barrier is basically done by these cells called as astrocytes. So as you can see here, it is forming a protective layer across the blood cells, I mean the vein and the arteries, and from them they will be taking only certain few compounds, and they will make it available to the neurons and the other cells of the brain, and that's how they nurture the cells. Okay, and on top of that, during the entire course of development, neurons का जो establishments है, connection establishments है, इस सब connection establishment के लिए they require guidance. The guidance comes from astrocytes. Okay, so basically they take part in parenting activities. Okay, and not just that, these astrocytes also provide a framework to the brain and the spinal cord. मतलब आपका जो brain का जो shape है, वो जो आप देख रहा है, वो neurons की वजह से नहीं है. Rather, it's because of astrocytes. These star-shaped cells are the ones which are providing the framework for establishment of the brain structure or the spinal cord structure. Now coming to microglia. Microglia, as you can see, these are the smallest cells available here. So what are they? As the brain is not having the flow of the blood into it, the defense molecules are not going to get in. So we require something to protect our brain from inside. And that is provided by these cells called as microglia. They are considered to be the modified versions of monocytes, one of the defense cells of the body. Then comes ependymal cells. Ependymal cells are the ones which will be these ones available on the linings, brain linings, on the spinal cord linings. So what do? They release this fluid called a cerebrospinal fluid, which is required for transportation. At the same time, this fluid is very, very thick, very, very viscous. Okay. So whenever we hit a target, let's say हम लोग ऐसे ही जा रहा है, let's say we are getting bombarded with a wall, or let's say we are bombarding with a glass or a pole. Okay. Usually we feel a bit of dizziness initially, but we get back to normal. Okay. So whatever these kind of physical shocks are there, all of these mechanical shocks tends to get absorbed by the cerebrospinal fluid. So it protects the sensitive part from all types of external shocks. Okay. However, there is a threshold value. Once this threshold value is crossed, then the destruction to the brain happens. Okay. Threshold value, matlab, let's say somebody is traveling on a bike and let's say they are meeting with an accident and they're falling on the ground with a very huge force. Or let's say somebody is banging somebody's head with a rod. Those kind of forces, उसके लिए mechanical absorption काफी नहीं है. So the threshold value crosses there, and that leads to damage of the brain. Okay, so minute shocks up to a threshold can be bore by this fluid called as cerebrospinal fluid. So we already have this structure called as cranium. ये जो hard shell है सर के ऊपर, that protects our brain, and beneath the cranium also you have a shock absorbing liquid. That is nothing but your cerebrospinal fluid, and that is released from these cells called as ependymal cells. Then we have the sixth category of cells called as satellite cells. These are discovered recently and their function is not yet known. Okay, people are still trying to understand what these cells do in the brain. I mean, not in the brain, in the peripheral nervous system. Then comes your neurons. The neurons are the typical cells of your nervous system. Okay, they are the main functional cells of your nervous system because they are the one which takes part in the communication. Okay, so a typical neuron has two structures: cyton, axon. Cyton means the cell body. Axon is the area of generation and conductance of electrical impulse. So this is a long tail-like structure called as axon, which is membrane-rich, which is lipid-rich region, and then you have a cyton. Cyton basically may or may not have these projections called as dendrites. So dendrites are the receiving regions of the stimuli. Okay, and inside the cytosol, you have all your structures like nucleus, endoplasmic reticulum, mitochondria, Golgi apparatus. On top of that, the neurons have extra structures called as nissl bodies. The nissl bodies will be acting according to the signals received, and they will be delivering certain factors called as neurotransmitters. These are the chemicals that are released by the neurons whenever they receive an input. Okay, and the same input has to be forwarded onto for communication. So for communication purpose, they release factors called as neurotransmitters. Okay, so the neurotransmission, once it is released, then the signal accumulation happens at this conical structure called as axon hillock. From here, the electrical impulse is generated, and this generated electrical impulse cannot flow continuously because of availability of these insulators, which are discontinuous. Okay, the myelin sheath. So how does the electrical impulse flow then? It flows in a jump manner. So basically it jump across the myelin and it will be landing onto this empty regions called as node of Ranvier. So we have many nodes of Ranviers. So across the nodes of Ranviers, the signal jumps, the electrical impulse jumps and they'll reach the end. Okay. So along with the electrical impulse, your neurotransmitters also travel to the end and they reach the axon terminal. This is where the signal delivery happens. So this is a typical structure of a neuron. Okay. However, if you're going to look at other types of neurons, there are four different types of neurons based on the structure. One are unipolar, which means that a neuron does not have any dendrite on it on top of it. It simply have access to the receptor region, followed by you have an area of conductance, and you have multiple regions of delivery. This is unipolar. Then comes bipolar, which means you have a sensory receptor, followed by you have one area of conductance, followed by you have the cell body, followed by you have another area of conductance. So you have two different uh, tails for conducting on either side of the so-called cell body. So this kind of a neuron is bipolar. Then comes pseudo-unipolar, which means you already have a single area of conductance only. Unfortunately, the body of the cell body is not located on in connection with it. Rather, it is located a bit away from this. So this kind of a neuron is called a pseudo-unipolar neuron. Then comes multipolar neuron. This is the one which has multiple projections on its cell body. So this has multiple regions of reception and multiple regions of delivery. So this kind of a neuron is called as multipolar neuron. So this neuron that we have seen here is a multipolar neuron. Okay, this is with respect to the structure. With respect to the function, we have three different categories. The ones that takes part in sensing or collecting the information are sensory neurons. The ones that takes part in delivering the outputs are motor neurons, and the ones that takes part in communication, मतलब सेंसरी से ले ब्रेन से लेके मोटर तक जितना भी कम्युनिकेशन होना चाहिए, all of the communication is done by these neurons called as interneurons. Usually the motor neurons and interneurons are multipolar in nature. The sensory neurons are either unipolar or bipolar or unipolar in nature. Okay, so these are the different types of neurons based on the structures and based on their functions. Then no, so we stop here for today. Stop here for today. The next functional components and all diseases and all we'll study it in tomorrow's class. Okay, so that's all for today.
Hello, sir. Sir.
Yes.